on everything.
Yeah, no, it was just the one goal, but yeah.
Oh, it was an on goal.
No, it was one goal, just like
Oh,
not
one.
much.
I
Yeah.
was going to say you're scoring on the wrong side then. If that's
Yeah.
that's still fine. So, what was the margin you guys won by?
We didn't win. It was just 1-1. I scored the first goal in the second half, and then they equalised at the end. It was a penalty. They got a penalty. So
Man, that was a tight game then. 1-1 for a full, what, 90 minutes?
it's 80 minutes, 40 minutes each way.
That is still a tight game.
Yeah, yeah. But I
Considering
could
how many
feel it.
goals you had,
Like,
just 2 goals. Wow, wow,
yeah,
wow. Good
yeah,
stuff, good stuff. Well, keep
yeah.
that
I felt
going.
very
Obviously, with
fit.
Liverpool winning, I hope you're carrying on that winning streak and scoring more goals. Good stuff.
Yeah, definitely.
Fantastic. So yeah, so that's fine. I swapped with your sister. We're gonna go a little bit on the dark side today. As you've seen, it's probably been a while since we did a little bit of this, so I thought let's just recap some of the few concepts, especially given now obviously in year 10. So we're gonna just make sure you're
Yeah,
consolidated on some of the previous principles you've already done in year 9, and if there's anything that needs brushing up, we'll brush up on that. So
yeah.
just to start us off, I thought we'll start us off with a little warm up. Okay, so just have a go at those 4, and then we'll get into some more biology.
Okay.
Now, while you're doing that, Jay, I'll just send off the video to your sister as well.
Oh yeah, thanks.
Yeah. While you're working through those.
I've got the first one, but I'm not sure about the other 3.
The 3, yeah, okay, don't worry. We're just gonna go through the ones that you know.
Okay.
So, did you get 1,000
Yeah,
for that?
1,000. Yeah,
Okay,
yeah.
good stuff, good stuff. So there's 1,000. Now, do we know what this little symbol means here? Do you know what this means? So it looks
Does it
like
mean?
this.
No, I'm not sure to be fair. No.
Not sure, that's fine. So this means micro,
Micro. Okay. So
okay?
micrometers. Yeah.
Micrometer is correct. And this symbol here is the symbol mu. Not sure how you spell it. I think it's mu. Instead of mu, but mu, okay?
Yeah.
It's got
Okay.
a little tail at the front and then the letter U as you would write it. Okay, so micro. So what does micro mean then? Can you remember any chance? Do you remember how many micrometers?
Um.
We can go
Is it
down
like
to, yeah, go on.
0.001 or something?
Nearly there. So if I had, I'm going to give you another one to try then. Okay, some nice easy ones. And then we're going to build from there. So hopefully you know what these are.
Yeah. Yeah.
Okay. Millimeter. How many millimeters will there be in a meter?
Um. Well, if we do the centimeters one first, that may be
Yeah,
easier.
that's fine.
So 0.0. Wait, hang on. So 100 centimeters in a meter. So 0.001.
0. Just 0.01, because because you'd say 1
sorry,
divided by 100,
0.0,
wouldn't you?
yeah.
Okay,
And then with the millimeters one be 0.001.
correct. Yeah, because you're dividing that by 1,000.
Yeah.
Good. Okay. So a micrometer is smaller than a millimeter.
Okay. Yeah.
Okay. And a nanometer is smaller than a micrometer. So if I had to write these in an order, okay, so then if I had to put an inequality sign, it would look like this. Okay.
Okay.
1
Yeah.
meter is bigger than 1 centimeter. 1 centimeter is bigger than 1 millimeter, which is bigger than 1 micrometer, which itself is bigger than 1 nanometer. All right. So the key question really is here is, what do these prefixes micro and nano mean? Milli, just to remind you, means anytime you see milli, it means if you change that to meters, you'll be multiplying that number by 1 times 10 to the minus 3. Can you remember your standard form?
Yeah.
Good, from your maths, well done. Okay. Centi means 100. So to change
So that
something
1 times
centi.
10. 1 times 10 to the minus 2.
Well done. Good stuff. All right.
Yeah.
The next guy now, micro. We times anything in micro by 1 times 10 to the minus 6.
Yeah.
All right.
Okay.
And nano. Have a guess for nano.
Would it maybe 1 times 10 to the minus 9?
Well done, because you know that word, a nanogon. Oops, you can't see that.
Yeah.
You know nanogon means 9-sided, so you're using that knowledge, so you're absolutely right, Jay. It's 1 times 10 to the minus 9. Okay?
Yeah.
Now, 1 nanometer, how many micrometers is that? Have a go at that one. So we've got all these conversions now. Try and see if you can work out number 4. Take your time.
Would it be like something like 1 times 10 to the minus 8? Or...
1 times 10 to the minus 8.
Because we know it's smaller than the micrometer.
Not quite. Yeah, you're right. So we know that 1 nanometer is smaller. You're absolutely right. So it's going to be 1 times 10 to the minus something, isn't it?
Yeah.
Because anytime something is smaller, here that's smaller, it's to the minus 9, smaller, minus 6. The nanometer is smaller than a micrometer. It's going to be 1 times 10 to the something, you're right. So what something is that going to be? Can we apply any mathematics to try and work it out? Any maths we can do? Sorry. Any calculations based the fact that we have these 2 pieces of information?
Could you
So maybe a little bit of a hint. So 1 nanometer we know is 1 times 10 to the minus 9.
Yeah.
And a micrometer is 1 times 10 to the minus 6.
Yeah.
So another way you could think about it is, how many times is this value here? How many times is 1 times 10 to the minus 6 bigger than 1 times 10 to the minus 9? Can't you think about that way?
How many times is it bigger?
Does it
Is
make
it
sense if we had to think about it that way?
would you do 9 divided minus 9 divided by minus 6 to work out?
Minus 9 divided by
Minus 6.
minus 6, okay, and what would that give you?
2/3.
Ah, nearly there. But because remember we're using powers, another thing
Yeah.
of course we could do, Jay, yeah, if we want to make life a little bit easy, well, we can say, how many micrometers are there in 1 meter? Let's try and think about it that way. How many
Okay.
micrometers are there in 1 meter? Can you answer that question? Let's try and answer that question first.
Okay.
How many micrometers are there in 1 meter?
1 meter. Okay. So to work that out,
Yeah,
would you do 1 divided by 1 times 10 to the minus 6?
correct. Good. And what does
Okay.
that give you?
Do you mind if I just go grab the calculator? Because then I'll be
Go
able
for
to
it. Yes,
understand.
Jay, take your time. Yes, I'll be waiting.
Cheers.
No worries.
So, and then times. And get an answer of 1,000,000. So would it
Hold
be?
on. So we say then you're right. We can write that down. 1,000,000. You can write it in standard form if you want. That's fine. But you can say 1,000,000 micrometers, Jay,
Yeah.
is the same as 1 meter. You happy?
Yeah. Yeah.
Now let's write the number of nanometers in 1 meter as well. How many nanometers are there in 1 meter?
1 meter. Well, 1 divided by 1 times 10 to the minus 9.
Okay, and what does that give you?
Sorry. Yeah, and then that. 1,000,000,000.
So we've got 1, 2, 3, 3, 3. And
Yeah.
this is nanometers. Yeah?
Yeah. Yeah.
So this is the same as 1 meter, and this is the same as 1 meter. So do you agree that I can therefore say that 1,000,000 micrometers is the same as that billion nanometers? 1,000 million nanometers. And I'd better zoom up because you can't see what I've done on the other side. So can you agree
Cheers.
that these two are the same? Because if that's 1 meter and that's also 1 meter, I can make these two equal.
Yeah. Yeah.
Yeah, which is what I've
Yeah.
done. So now hopefully you can see that 1,000,000 micrometers is the same as 1,000 million nanometers.
Yeah, because you can write that because they are both equal to the same thing. They're both equal to 1 meter. So
Correct, absolutely correct. But now actually what I want to find out is, well, how much is 1 nanometer? worth in terms of micrometers?
Well,
Now,
would you
can you
do?
remember that little trick of cross multiplying? So can you
Oh,
see
yeah.
on this side, I've put all my values for that are in nanometers. And this is the side where all my things with micrometers are going to be. Oops, sorry. Not a good line, that. And this is where all my micrometers are going to be. That's the reason why my 1 nanometer has been put this side, because it's the nanometer I want to work out. You happy?
Yeah. So
Good.
could you just do
1 times that.
yeah, and
One
then
side, that times that. So obviously, whatever multiplies with a question mark is the thing I have to divide by. So
okay.
1 times a million.
Yeah.
Yeah. So it's going to be 1 times 10 to the 6, which is equal to 1 times 10 to the 9 multiplied by a question mark. Because this question mark is going to be my value in micrometers.
And then you move over your 1 times 10 to the 9.
Good. What does that give you then? Tell me.
So 1 times 10 to the 6 is 1 times 10 to the 6. So
Good.
then we got 1 times 10 to the 6 is equal to 1 times 10 to the 9 times your question mark. And if you
So
move
we're
it
going to work
over,
out what the question mark is.
yeah, so do you do 1 times 10 to the 6 divided by 1 times 10 to the 9?
Absolutely.
And you get 1 times 10 to the minus 3.
Absolutely. Spot on.
Yeah.
So you knew it would be a minus, you just weren't sure what it was going to be. Yeah, you didn't know whether it was a -4, a -5, or a -6. But hopefully, if you should ever get lost and can't remember the conversions, you can just use some maths and make that your starting point. Okay?
Yeah.
What you absolutely have to try and remember is that anytime you see micro, to change it to simply this unit alone, you have to times by 10 to the -6. Same even if I had 1 microkilogram. It means to change this to kilograms, it's 1 times 10 to the -6 kilograms.
Okay, yeah.
Okay? That micro means times 10 to the -6. The nano means times 10 to the -9. All right? Good.
Okay.
So now that we've done those, write them down if you, oh, sorry for that, phone shouldn't ring.
No, don't worry.
Now, what I'll suggest then, Jay, is have a go at writing any of these useful ones. Just write them down because I'm going to rub them, especially the ones that you think you might want to remember more.
Okay.
Hi, man, I'm teaching at the moment. If you could call me maybe about in the next hour and a half. Hello? Sorry. Okay, and this is also a pretty useful one. How many nanometers to the micrometer?
Yeah, okay.
Okay.
3 equals 2. Yeah, and then I know the 1 millimeter equals to the meter. And the
yeah, you know what that one is, isn't it? And you know the centi one, so you don't have to worry about those ones. Yeah?
Okay, that's good, yeah.
Good, man. Good stuff. Okay, so we're going to be using some of this information today.
Yeah.
And the only area really we're going to use it because in biology, they will require you to remember those little prefixes. And there's another subject where they want you to remember those prefixes as well. Which subject do you reckon that is?
Would it be maybe chemistry?
Not so much in chemistry. They don't try and catch you out with those units, but there's another one.
Would it be physics?
Absolutely. They love that. Physics loves it a lot more than biology. Okay, so before you go into your physics exam, make sure you look at those little prefixes because in physics, we've actually got more. We've got mega, we've got giga, yeah? So there's a lot more in physics. But when we do
Yeah.
physics, we're going to try and be doing some of them as little starters. So
Yeah.
this neatly brings us into this new topic we're going to look at.
Is it
And that is,
microscopes? Yeah.
oh, how did you guess that? It can only be
Visual.
microscopes in biology, isn't it?
Yeah. Yeah.
Okay. So we might fly through this if you can remember a lot of the concepts, okay? But
Yeah.
we'll build on this and then go into some of the basics that we may have done, and then just have that as a foundation to start doing a little bit more biology. So microscopes then, Jay, tell me, what can you remember? How many different types of microscopes do we have?
Is it two? And is one like a laser one
Yeah, similar. It's been a long time, eh? That's why I said we've
yeah.
got to visit the dark side, as I call it.
Oh, yeah.
All right, so light microscopes. Okay.
Yeah.
I've got my reasons for calling it that. The only reason, it's not that I don't like biology, it's just the fact that they are they have a lot more things compared to physics, which they can't fully explain. So they have to come up with theories like evolution and all these things.
Okay.
That's why I say it's a bit of the dark side. Anyway, so we have light microscopes, and then there's another one as well, which is known as an electron microscope.
Oh, yeah. Yeah.
Okay,
Yeah.
good stuff. Now, of course, a lot of this stuff was built by physicists, so we love biologists.
Yeah. Yeah.
So light microscopes. So what can you remember about light microscopes? Let's just try and run through some historical accounts of when this stuff came about. You'd never be asked that really, as long as you remember that the light microscope came before the electron microscope. That's all you need to know. But to add some detail, this guy was invented in about the 17th century. Okay, was electron microscopes only came in about the 1930s. Okay, that's the key thing. Now, light microscopes, what do they use to be able to see their image? Yes, go on.
Aren't they cheaper than like the electron micro as
Indeed, they are way, way, way cheaper, and that is why they have the school has allowed you to use them, and actually that's what your school would normally have. But if your school has an electron microscope, it's universities that have electron microscopes, okay, and not all universities, so they're pretty expensive. So this light microscope uses light to form the image.
Yeah.
And you're absolutely right in terms of price, they're cheap. Cheap and light as well. They're not heavy. Yeah, they're portable. So I can say cheap and portable. Okay?
Yeah.
Electron microscopes, on the other hand, are very expensive. And what they use is they use electron beams. Uses an electron beam to form the image. Okay? Now, as we said, they are expensive. Okay?
Yeah.
Expensive, and they're also large.
Okay. Yeah.
And also, you need certain special conditions, need special air pressure, humidity, and temperature. So the conditions in which you can actually do the viewing have to be controlled.
Yeah.
So light microscopes are good for looking at, if you like, quite a few cellular structures, but not very good at looking at what we call subcellular. So what I mean by subcellular, as in the tiny parts of the cell. So looking at things like chromosomes, which are much, 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 much tinier than, say, a nucleus. So you can see a nucleus with a light microscope, but then to see a ribosome becomes very, very difficult. Why? Because the light microscope, if I just come down here, so light microscope only has a maximum magnification, magnification. of approximately 2,000 times. So you will see this expressed as times 2,000. Okay?
Yeah. Yeah.
Like so. All right?
Okay.
So at this level of magnification,
Mm-hmm.
if you use the microscope, just to try and understand it, if you put me under a light microscope, I
Yeah.
would be about, I'd look about 3.5 kilometers big.
Wow.
Yeah.
Yeah. Yeah.
So
Okay.
at that magnification, sorry, Jay,
Um,
I'm teaching at the moment. Do you want to call me in about an hour 45 minutes? I won't finish until at least another. Yeah. All right. Right. Okay. So about 2,000 times. So all I've simply done now, we're talking about this concept of magnification. Yeah. So we're
yeah.
going to delve deeper into that. But before that, we're just going to look at also the electron microscope. Now, of these types, you've got two types of electron microscopes, Jay.
Yeah.
Okay.
Okay.
You've got a tunneling electron microscope. And also you've got a scanning electron microscope. Okay. Now,
Okay.
let's just think about the magnification of each of these. Now, generally, an electron microscope can magnify up to about 2 million times. Okay. So you can see there's a huge order of magnitude in difference. This is magnifying 2 million times. That's only magnifying 2,000 times. So if you had to look at me in electron microscope, it would be whatever my height is multiplied by 2 million. So there's a lot that you can be able to see. Okay, so that would be about 3 million meters, which whatever that would be in kilometers is a pretty huge number compared to me
Yeah.
being just 3.5 kilometers.
Yeah. Yeah.
Okay, good.
Yeah.
So tunneling microscope, scanning electron microscopes, these give 3D images. Let's move this. Let me zoom out. These give 3D images.
Okay.
Whereas tunneling electron microscope, these give 2D images.
So would the scanning one be a bit more expensive than the tunneling one?
Yes,
Yeah.
it's going to be a little bit more expensive. You're absolutely right. Yeah. But there's a little bit of a subtlety actually in that the tunneling electron microscope
Yeah.
gives you far greater magnification than the
Ah.
scanning microscope.
Okay.
Okay.
Yeah. Yeah.
So this here, even though both of them will magnify to a great value of about 2 million, the one that has got the greatest magnification of these is the tunneling microscope.
Okay.
Okay. So this has got the, I'm going to use a different color here. My board is getting absolutely overloaded. Greatest magnification of all of them.
Yeah. Okay.
Makes the image look the biggest. So the other thing we're going to talk about is just this formula for magnification. What do we mean by magnification? Can you remember what the formula for magnification is?
I don't know the formula. No.
Okay.
No.
But maybe you might think about it intuitively. If
Yeah.
I had, I don't know, the picture of, let's think of something, a marble. So the actual marble, this is my actual marble, is 2 centimeters. And I take a huge magnifying glass. And under the magnifying glass, it now appears to be 10 centimeters.
Okay.
But how much have I magnified my image?
Would it be 5 lots?
By 5 lots, you're right. So you can see that my magnification is times
Time.
5.
Yeah.
So remember, this is my, sorry, I beg your pardon, my object, and this is my image.
Yeah.
So what
Yeah.
did you do to find that 5?
I did 10 divided by 2.
Yeah. Image size divided by
The object
object
size.
size.
Oh,
So
okay.
image size. So you know you can do it intuitively. It's just if you have to quote it as a formula, it's also worth remembering. Image size over object size. Okay? And that's what we mean. So we should be able to rearrange this formula, but you shouldn't have many problems because I think you will be fine with that. Okay?
Yeah.
Another thing that we realize about these microscopes is the type of samples that we can view in each of them. With a light microscope, you can view live samples.
Oh, okay.
Okay?
The electron you have to not.
You have to kill them. They have to be dead.
Oh, okay.
Because the electrons use a physics principle known as diffraction. So normally, if you probably don't think we've done waves, did we do waves? Yes, I think we did waves. When a wave passes through a gap, it spreads out.
Okay.
Yeah? And all waves do that. When they pass through a tiny gap, that's about the size of the wavelength of the wave, they spread out. And it's these, as they spread out, they form shadows. So it's these shadows that form the image. So if you've got particles in a cell and they're moving around, it's very hard for the electron beam to actually spread out and form a clear image of what's there. So the stuff has to be static. It has to be rigid.
Okay, yeah.
So if you ask perhaps a disadvantage of electron microscopes, we already know to say, well, they're expensive, they need special conditions. There's another downside in that you need to have dead specimen for you to look at. And if you're trying to look at something that's live, it's impossible to view something that's live with an electron microscope.
Yeah.
But you can do that with a light microscope. Okay?
Yeah.
So now that we've spoken about magnification and realized the importance of this equation, we're now going to look at something known as resolution.
Resolution. Is that actually
Can you remember what resolution is all about? What is resolution all about? So when I say resolution, I want you to almost think about resolution and magnification as being very, very closely related. So anytime you talk about magnification, in the back of your head, think resolution, because you can use it as a second point if you're ever writing a 6-mark question about the differences in the microscope. So there's not only a difference in magnification, but there's a difference in resolution. So what is resolution? What do we mean by resolution?
Is it how clear a picked image is?
Yeah, pretty much there. Pretty much there, actually, Jay. So let's think about an instance where you've got a car. Far away, it's a dark night. Now, do you agree? Say, imagine this has got headlights and there's light coming out. Do you agree that when this car is very, very far away, all you're going to see is one bright blob, one bright light? Do you agree that when the car is very, very far away, you would not be able to make out that there's 2 headlights coming at you?
Yeah.
The further away the car goes, the harder it becomes for you to distinguish that it's actually a car.
Yeah.
If somebody said, is that a motorbike or is that a car? You would have no way of knowing after a certain distance, because your eyes will not be able to make out that that light is coming from 2 separate sources. But as that car gets close to you, okay, my version of my car, as that car gets close to you, you will then make out to say, hang on, that's actually a car. It's not a motorbike.
Oh, yeah.
So that is what we mean by resolution. The ability to be able to make out 2 objects as separate.
Okay. Yeah.
Now, we know that eagles have got very good eyesight, better eyesight than humans.
Uh-huh.
Yeah. So if we're
Yeah.
if we're thinking in terms of resolution, if I had an eagle looking at the same car where I could only see one light, the eagle will actually be able to see two headlights. It will know that's a car in the distance. I
Yeah.
won't. Similar thing happens with microscopes. So when you say resolution, ability to
Yeah, 'cause
distinguish.
whenever you look far away and there's a car, it always looks like a motorbike when it's
You
miles away.
remember as well now, don't you?
Yeah, yeah,
When it's at a certain distance, absolutely right, absolutely right. But of course, if you had something like a binoculars, what happens? You're quickly able to distinguish
yeah,
because you can zoom in that much more into the road.
yeah. Uh-huh.
So a binoculars improves your resolution.
Okay.
Why does it improve your resolution? Because it magnifies
Yeah,
what you're looking at. So that is why I said, whenever you think of resolution, think of magnification.
yeah, yeah, yeah.
The bigger the magnification I've got, what happens to my resolution? The better
Yeah,
my resolution becomes. So resolution is ability to distinguish two separate objects as separate. Okay?
yeah,
Now the resolution, now we need to think about resolution in terms of light microscopes. And also in terms of electron microscopes. I'm actually going to split this into the transmission, tunneling, and scanning electron microscope. Okay?
yeah.
I just call this a transmission electron microscope. Make life easy. They're the same thing, really. So for light microscopes, the resolution that it can give me is not as high, which is what we've said, as these, because these guys have got huge magnification. You happy with that?
Yeah.
So a light microscope can only resolve to about 200 nanometers. So if
Okay.
those two objects are 200 nanometers apart, then a light microscope will be able to tell that those
Yeah.
are two separate objects.
Okay.
But if these objects are less than 200 nanometers apart, a light microscope will make it just look as if it's one object.
Okay. Yeah.
Okay?
Yeah.
Now, a scanning electron microscope, this has got a
Yeah.
resolution of 0.2 nanometers. Now, look at the difference.
It's not as good. Like by a huge amount.
Yeah. And the
Yeah.
transmission, sorry, I've got them wrong. Transmission is 0.2 nanometers, and this
Okay.
is 10 nanometers.
Okay. Okay. It's a bit better.
So
Yeah.
remember what I said? I said a transmission electron microscope has got the greatest magnification out of them all.
Yeah. Uh-huh.
And look, it can resolve things that are very, very, very close.
Mm-hmm.
Followed by a scanning electron microscope, which can only resolve to 10 nanometers. And finally, a light microscope, which resolves to 200 nanometers.
Yeah. Yeah.
Okay? So those are the key elements there that we need to cover in terms of microscopes. But there's one thing that I would like us to cover, and that is, again, love testing this. I've seen 3-4 mark questions on them. Love to cover, how do you prepare a slide so you can have a look at, how do you prepare the slide of a microscope so you can actually look at a specimen, okay? So we're going to look at that to some level of detail. I'm just trying to think if there's anything else I need to add. So we've got the formula for magnification. I told you that guy can produce 3D. This only produces 2D. Yeah. And I also mentioned the fact that this can be used to view live or dead specimen. Whereas this here, these two, whatever it is you're viewing has to be dead. You remember that?
Yeah, yeah.
Yeah, good stuff. Okay. So what we might try and do then is just undertake a few calculations, shall we? Just to
Yeah.
test this idea first, make sure that we understand this idea of magnification, and then later on we can go into a few more details of uh, how to answer some 6 mark questions. All right.
Yeah.
Let me just where are we? Yeah, that's fine. Just bear with me a moment, Jay. Close. Now, let me just look here. Just one moment, Jay. Thought I had this out here ready. Should find it. Nope, gone too far. Trying to look. No, it's not that one. What is this one? This one? So I have to share it from my iPad, Jay.
Okay.
Yeah. I thought I had it in a folder, but I've got it somewhere else. So I'm gonna have to share it and just bring that up. So photos. Photos. Not that one. This one. Okay, cool. I've got them. And screen mirroring. And server. There we go. Yes. Just taking its time, Jay. It will load up very shortly. Go on then. All right. Great. Okay, so new share. Okay, hopefully you can see that, Jay.
Yeah.
All right, great. So let's just have a go at those questions.
Yeah. Do you know how you could change the focus of things? Is that the resolution as well, sort of?
Yes, you're absolutely right, Jay. That's very good. You've tied that into it. Shows our understanding there. So while when you are playing with the focus button, what you're essentially doing is you are improving the resolution. Yes. And there's one more thing I meant to mention, and that is how to find the overall magnification if you've got two lenses joined together. But I'll let you do some of these questions. I did think I was missing something there, but you're absolutely right, Jay. Spot on.
Yeah. Microscope. It's cheaper. Or you can produce live photos or live images.
Yeah, you can view live images, correct?
In an electron microscope, there's a larger magnification.
Larger magnification, yeah. And also you can mention better resolution, higher resolution.
Yeah.
Okay.
And then 2A.
So hang on, Jay, for that one, just to check what you've done, make sure you've answered the question. You have stated
one
one
disadvantage.
advantage and one disadvantage.
Okay.
Okay.
Yes.
So A of a light microscope. So it's a 2 marker, so they're going to be expecting 2 points. One advantage and one disadvantage. And then same with part B for the electron microscope. They want one advantage and one disadvantage.
Yeah.
Okay, good.
Okay, and then for a light microscope, the disadvantage is um, um,
Think about the opposite of part B of what you've written.
could I just say not as good magnification? So
Low
low.
magnification, yeah.
Okay.
So low magnification compared to an electron microscope, for example.
Yeah.
Or you can say lower resolution than electron microscopes.
Oh yeah, resolution. Yeah. Yeah, and then a larger for the B an electron microscope. You could say it's not portable.
Yes, absolutely. Yeah. Not portable, or you can say very expensive. So they're
Yeah.
all, those are the solid points.
Yeah.
Not easily portable, at least anyway, without saying not portable, not easily portable.
Yeah.
Okay, two part A. Now, with this one as well, Jay, just a little piece of advice. Okay, what I would go for if they ask me for one advantage and one disadvantage, I'd definitely go for the big ones, the big advantage and the big disadvantage. So cheaper
Yeah.
is a big advantage. Low magnification, definitely good. This one here now, part B, larger magnification is definitely an advantage. The other one as well that you can say, because if it's cheaper, you can say very expensive. So think
Okay.
about it as the advantages that are with the light microscope are the disadvantages of the electron microscope and vice versa.
Yeah. Yeah.
Does that make sense?
Yeah. Okay.
Okay, good.
And then 2A. A student measured the diameter of a human capillary on a micrograph. The image measures 5 millimeters, and the student knows the magnification is times 1000. How many micrometers is the diameter of the capillary?
So this is where you might need some of those conversions now, Jay. And
Yeah.
you might also need that formula for magnification. Can you remember what the formula for magnification is?
Isn't it the object size divided by the... I know how to do it, but I can't remember the formula. Object size
Okay.
divided by the
So it's the
magnification.
image size. How big is the image? How many times bigger is the image
Yeah.
compared to the object? In other words, it's the ratio of the
Yeah.
size of my image to the size of my actual object, the actual size of the object, without
Yeah.
using any magnifier to look at it.
Okay.
Okay,
Yeah.
good.
Is this the final answer?
Oh, well, you've given me the answer in nanometers. The question wants the answer in what?
Oh, micrometers.
Correct.
Oh, 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 yeah.
Okay. Sorry, I should have spotted. I thought you were just using a different conversion. You may have misread it, but it is micrometers.
Okay, okay. So the micrometers is this one, the
Okay.
UM.
So I wanted with the mu m, that little symbol, correct? Micrometers. So how many 5,000 million nanometers, how many micrometers is that?
Okay. Oh, wait, hang on. Can we just convert this using that?
Okay. And with this one, Jay, you're going to have to
Yeah,
use the formula, aren't you? The formula for magnification.
yeah,
Okay, have you used that? So magnification equals the image size divided by the object size. So what they want to say is, what is the actual size? The actual size of the object that you're measuring before you look at it with your, before you magnify it. What would its actual size in micrometers be? Does that make sense?
yeah. So would you divide it by 1,000?
You
Not
have
time.
to, yes. So you have to
Yeah,
divide 5 millimeters by 1,000 first. And that will give you the number of millimeters, its actual size in millimeters.
yeah, yeah.
Is that what you did?
So no, I didn't do that. I times it by 1,000, not
No,
divided it.
you have to divide it. You have to
Yeah, yeah,
divide it because your formula is magnification equals image size divided by object size.
yeah,
Okay.
yeah.
So to find the actual, oh, can I? Maybe I can. Oh, yes, I can. I'll do that. Do it that way, and I'll share both screens. Okay. So this is what we're using here, Jay. So they've given you the image size.
Yeah, yeah.
They want you to find the object size, actual
Mhm.
object size. And the magnification is 1,000 here.
So then you rearrange it so that object size is the subject.
Good. So it's object size equals image size over
Right.
magnification.
Yeah,
Okay,
yeah.
good. So I'll come out of that so you can see the whole screen. Good.
Okay. And then, so I'm just trying to think where to start if I can.
All right, so let's do a part of it together if it helps, and then you can take it away when you feel you know where you're going. So the image size is, how big is the image size?
Its image size is 5 millimeters.
Well
So
done. So I'm going to put
yeah,
my 5 millimeters there
yeah.
and divide it by, what's my magnification?
Magnification is 1,000.
Well done.
That'll be 0.005.
Okay, and what would the units be then, Jay?
It'll be um
So
Would it
now
be still
think
be
physics here, Jay. If the top one is in millimeter and the bottom has got no units, what would my final answer be in?
millimeters minus 1?
Not millimeters minus 1, no. The units of these at the bottom here
Yeah.
are the same as just a 1, because there are no units there.
Okay, so would it just be millimeters?
Absolutely. Millimeters divided by 1 is millimeters. So this is the size of the object.
Yeah, yeah.
Now all I've got to do now is convert this here into something that has a micrometers at the end.
Okay.
You happy with that so far?
Yeah, yeah, yeah. So we use the conversions to do that.
Okay, go for it.
Yeah. 0.005 millimeters, and they want it in micrometers.
Micrometers, yeah, correct.
So we know the equation for that is if we use the meters. So, yeah. 1 times 10 to the minus 6 meters. Times 10 to the minus 3 millimeters.
Okay.
And then we've
You're
got
doing well. Keep going, Jay. You're doing well.
millimeters, and then
Good.
divide. And then is it just 5 micrometers?
Well done, top man. Excellent, Jay. So you have got the final answer in end, and it's 5 micrometers. Okay. So it is 3 marks for a reason, because you have to get to use a formula. Then you've got to do some conversions. All right. So you're absolutely right. It is 5 micrometers. Now there could have been another way to think about it the moment you stopped here. Once you get there, you know that anything with milli, that milli means, to change it to meters, that milli means it's the same as times 10 to the what? Minus 3, isn't it?
Yeah.
And this would be in meters. So if I convert this and move this 1, 2, 3, it means I can rewrite this as 5 times 10 to the minus 6 meters in standard form. I can write that as this.
Yeah, yeah.
And then if you remember, this here can be replaced by the symbol micro.
Yeah.
So then you simply get your 5 micrometers, a much quicker way to do it. But that's as you get more and more confident with the prefixes.
Yeah, yeah.
Okay? It's all going to come together and piece together. Okay, next one, that should be nice and easy for you, Jay. Part B.
So, part B, yeah. Okay.
Do you need a reminder on the formula, Jay?
Yeah, I think it's 800 divided by 20.
Correct.
Yeah. I was just trying to double check it.
Yeah, good.
20, and then 80 or 2, 40. Yeah.
Spot
40.
on.
There's no units, it's just
No,
times
there's
4.
no units because the two cancel out. Okay, so you're right. It's just times 40. Well done. Okay, good stuff. Right. Good, 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 Jay. So what we're going to do then, Will, I think it's definitely a good time to pause a little bit
Okay.
after that, and we'll pause for about 5 minutes. It's now
Yeah.
18:04. So if we meet up at about 18:09, how's that?
Yeah.
Yeah,
That's good.
great stuff, Jay. So I'll see you very shortly in the next 5.
Okay. All right. Thanks. Cheers.
I'm recording, but it's fine.
Oh,
We've got the
don't worry.
points. We've got the points there anyway in relation to number 3. Okay. So
Mhm.
when you looked at the onion cells, can you remember what you did, Jay? Because this is again something they like asking. How would you prepare a sample
Yeah.
to be viewed under a microscope? Can you remember what you did?
So we get a, is it a slide, a glass
Yeah,
slide,
you got
and
a
then
slide. Well done. That is the right name for it.
we'd wet it a bit
Yeah.
and put the onion ring on top of it, and
Okay.
then
Now you'd have used a tweezer to take a
yeah,
thin layer.
yeah,
Yeah.
uh-huh,
As opposed to a huge chunk of onion. You didn't put a huge chunk of onion there.
no, it's
You use a
thin, yeah,
very thin layer.
and then we'll be sandwich it with another slide.
Nearly there. There's another important point that there's something else that you did. You took a little pipette and you grabbed some iodine
Oh yeah,
and you put a
yeah.
few drops of that.
Isn't that dye the color of it?
Yes.
Yeah.
So it's a stainer. Okay.
Mhm.
So iodine. Don't forget that part because a lot of students forget about it. You need something to stain your sample so you can
Yeah.
actually look at it. If you just use without the stainer, you won't see anything.
Yeah. Yeah. Okay.
Okay. So once you've got that, and then you take another little piece of glass. Now this has got a name. It's called your cover glass.
Oh yeah. Okay.
Okay. And you put it there and you sandwich your sample between the two and you are ready to go and analyze it under your light microscope.
Yeah. Yeah.
If they say you want to look at cheek cells, again, this is something you may have done when looking at electron microscopes. Maybe they give you a little swab and you
Yeah.
swab the inside of your cheek. I don't know if you did that.
We didn't do that one, but I know other classes did, but we didn't actually get to that one. We just did it.
Okay. Your biology teacher was probably as brave enough to allow people swabbing their mouths, but there we go. So you get the cover glass, the slide, very similar,
Yeah.
and then you take your swabbed sample from your cheek
Yeah.
and you then, if you like, rub it against this slide. Yeah.
Yeah.
And then
Okay.
you take a stainer. Now, the stainer you use is one known as methyl blue.
Okay.
Very similar again, a few drops, and then you put your cover. If you can't remember the names of the stainer, whether it's iodine or methyl blue, just say, add a few drops of liquid that will allow it to stain.
Okay. Yeah.
Okay, if you can't remember the names, because different things require different stainers. And
Yeah.
instead of you remembering all the different types of stainers, they're not going to be testing you at that level at this stage. Okay.
Yeah. Mm-hmm.
All right, great stuff. So it's really good that we've practiced on how to answer 6-mark questions because
Yeah.
they do take quite a bit of practice when you are doing biology, especially. And one of the things I say that's worth practicing in biology is more answering these 6-mark questions, because generally, if you can answer these 6-mark questions to a good standard,
Yeah.
you're on a
Yeah.
grade 9.
Okay.
Okay.
Yeah.
So what you want to have is you want to have a good strategy, and you want to make sure that you understand the command words, because always it's the command words that students miss out on.
Can you do it
Look at
in
it.
bullet?
Oh, electron microscope and light microscope. Oh, I know what those are. Oh, I'm just going to write the advantages and disadvantages. But actually, it's
Yeah.
best to take time and say, well, what does it actually say? Evaluate the use. Stop there. What does that mean? Then think
Compare.
about that question in a different context if you're not sure. What if somebody asked me to evaluate? I gave an example of a game that was played yesterday. What would come to mind? or if somebody said, evaluate the use of a spoon compared to a fork,
Yeah.
what would
Yeah.
come to mind? Yes?
Mhm.
So I might start thinking, okay, well, a fork has got these features, which does not make it very good for drinking soup, for example. Yeah?
Yeah.
So it's the use. So once you get that, anyway, with practice, we'll get there, Jay.
Yeah.
Okie dokie. Any questions so far, Jay?
Can you use bullet points for every single
6
subject
marker.
line?
Yes.
Yeah. Even
At
for
this
like,
present stage, yes. When I was in school, Jay, they used to sometimes be specific, and you might see some old questions from some old past papers which say you will be given a mark for the quality of your English and your grammar.
yeah.
In those cases, generally, you were better off not using bullet points, but actually writing full sentences, everything in full sentences, and making sure it was grammatically correct.
Yeah. Yeah.
But then they realize it's a bit unfair because you do have people who come to this country, maybe in year 10 from other countries,
Yeah.
who can't necessarily speak English very well and can't write well, but it doesn't mean they don't know the science. They know it very, very well because at the schools they would have been at in other countries, they learned it, but they learned it in either Arabic or a different language, and they can't quite translate it all the time. So it's much easier for them to write short sentences which are bullet points.
Yeah. So they
But just
get
to
the
kind of
point.
make it fair. So at this stage, they are more interested in the points you make. They're not even going to give you marks if sorry, they're not going to penalize you if you spell something incorrectly. As long as if you read it, it kind of reads like it. Yeah.
Yeah.
So you might say, I don't know, cells, and you write it like that. They
Yeah.
know you mean cells. Okay.
Yeah.
So they're a little bit more lenient, but obviously if you know the spelling, by all means write it correctly, because then you are setting yourself apart and showing them what caliber of students you are from the very onset. All right.
Yeah. Okay.
Great stuff. Great stuff. So what we're going to do then, Jay, is we'll try and look at some actual exam questions and just see what kind of stuff these guys have.
Yeah.
Says my screen sharing is paused. How do I go back to, oh, I know. If I go there, cell. All right, so these are actual exam questions we're looking at now here, Jay.
Yeah. Okay.
All right. So there we go. Starting off with number 1.
Okay. Well, it's not 3D, it's 2D.
Yeah, good.
It's times 200. So is it one of the light microscopes?
Absolutely, because we know that a light microscope will magnify up to 2,000 times.
Yeah.
Yeah, and this is less than 2,000. So we're going to go for a light microscope because we can also see that it is not a 3D type of image. So it's a type of
Yeah.
image we could see under a light microscope. Okay, well
Yeah.
done. Good.
And then B.
It's a bit of a nasty one, this, Jay, because you won't be able to do it because I just see that you can't measure the real length X, can you?
Yeah.
Yeah, you'd need to know that. Not to worry. We'll go for the next one. Let's go for that one then, Jay. Number 2. Oh, again, this one. Sorry. No, not good, not good. I know which one we're gonna go for. We're gonna choose this one here. because you don't need to. The others you can only do once I email them to you, Jay, because then you'll be able to print them off and actually take measurements off it. Okay. I think
Okay.
it's a bit unfair if I ask you the scale bar. Use a ruler. Yeah, we can't do that one. Definitely can't do that one. All right, number 4. Yeah.
Okay.
The diagram
Yeah.
shows 2 cells, a bacterial cell and a plant cell. I'm not going to ask you to answer what is the function of a ribosome because we haven't yet really touched on that. But
Isn't
you
it
can
to do
see
with
the
oxygen?
magnifications. Do you know what a ribosome is? Do you want to try on that?
Is it to do with oxygen, or is that
Nearly there. You're so nearly there.
Yeah. Is it to energy? No.
So that's mitochondria.
Oh, okay.
Ribosomes are for making proteins.
Oh yeah, I knew it 'cause we did it at the start, right at the start, but I forgot kind of a bit. We did when we were doing cells.
Yes, yes, yes. Good, good. So coming back anyway. It will come back. This is more a
Yeah.
refresher more than anything.
Yeah.
Okay. Now it says both cells are drawn the same length, but the magnification of each cell is different. The real length of bacterial cell is the real length of the bacterial cell. Now this is our bacterial cell. Can you see?
Yeah.
The real length, it says, is 2 micrometers.
Yeah. And then the
Now it says calculate the real length x of the plant cell.
okay.
So that's my plant cell.
Yeah. Yeah.
And they want to find the real length.
Okay.
And can you see the magnification here, Jay?
Times 500.
Times 500. Good. So we're told it's times 500. So the real length of a bacterial cell is 2 micrometers. Calculate the real length x of the plant cell. Give your answer in micrometers.
Yeah. Yeah.
Show clearly how you work it out.
So would we do 2 divided by 50,000?
Okay, so 2 divided by 50,000.
Yeah.
Now, let's just be aware here. So let's write down our formula for magnification so we don't go wrong. Yeah?
Okay.
Just
So
make sure that we're doing the right and making the right thing the subject of the formula.
magnification
Yeah?
equals to object size
Okay?
divided by image size. I think, or is it the other way around?
Now just think about it. When you magnify something, what looks bigger? Your image or your object?
Your image.
Well done.
The image divided by object.
Well done.
Yeah.
When you magnify it, you're making it bigger. And you know that magnification would normally be a number that's bigger than 1.
Yeah. Yeah.
Good.
So it's going to be 50,000 divided by 2.
Now, what are you trying to rearrange there, Jay? What are you trying to work out?
So we know the magnification.
Good.
We know the real length. So we know the object size. So we're trying to work out the image size.
Okay, good.
The OS times magnification equals
Okay.
to image size. And then so we just do the object size times our magnification.
Okay.
So 50,000 times our 2 micrometers. Yeah.
Okay. I'll bring this up because obviously you can't. Yeah.
Yeah, thanks. And then that is 100,000. And then we know now that the image size is 100,000 and it says they're both the same.
Yes.
So
both image sizes are the same.
So now we want to make the object size the subject of the formula.
Yes, because you want to work out the real size of it.
Yeah. Divided by object size. So image size is 100,000.
Okay.
Divided by. I'm sorry. So original size equals to image size time divided by magnification. 100,000 divided by 500. 1,000 divided by 5 is 200.
Okay.
Equals to 200. Is it? I can't remember. Is it
Okay.
micrometers?
Now, if ever you're confused, Jay, look at the units that you're using in your values, in your numbers. That 100,000, what are the units in?
Is it micrometers?
Yes.
Yeah.
If that one
So
top
the
is at micrometers, that one is going to be in what?
micrometers as well.
It's got to be in micrometers because the 500 is just a magnification. It's got no units. So all your
Yeah.
units are going to come from the top. Okay.
Yeah.
All right. Not to worry about that one. I'm not going to worry too much about that. That is absolutely fine. Okay, so I'll let you have another go, Jay, at, or rather not another go, but I'll let you have a go at the questions in this paper once you are able to print them. Okay,
Yeah. Okay.
nice one. Okay, so we are going to move on to cells a little bit and recap on that and see what we can remember. This and give you a new share. Here we go. Cool.
Yeah.
So, quick recap on cells. And with this, we're going to be looking at both animal and plant cells. Okay? So now, Jay, animal cell. We're going to annotate it, and you're going to tell me the names of the different parts.
Sorry, so you broke up then for a bit, so I didn't hear
Okay,
what
so
you.
we're going to draw the cell. We're going to
Yeah.
annotate it. Think about the role
Yeah.
of what each part plays. Okay?
Okay.
Just as a little recap. Okay.
Yeah.
So
Okay.
there we go. This is my animal cell. Okay,
Okay.
and right. So let's have a go at labeling what's in here now.
Yeah.
I've
Okay.
exaggerated these, so I'm going to exaggerate this.
Yeah. So
Okay, so when
is
you're ready,
that
Jay?
is that big circle with the lines through the nucleus,
Yeah, indeed.
and then the cell wall is the blue line going around.
This one. So I'm looking at an animal cell first. Okay,
Sorry, say that
so what
again.
I've drawn is an animal cell. So we're going to do an
So,
animal cell first, then we're going to draw a plant cell. Okay, so I've got a nucleus, well labeled. What's that next part?
wait 1 second, sir, because it keeps breaking. I'm just going to put my
Oh, does
data
it?
on it. Yeah, I'm just going to put my connect my hotspot to it so then it doesn't
Okay.
wake up. Keep doing it.
Any better? Hi, Jay. Can you hear me?
Can you hear me? Sorry.
Oh, yeah. Perfect,
Yeah. All right.
perfect. Okay, cool. All right, we're all up and running then. Better connection now?
Yeah, better connection now. Yeah.
Great. Okay, so where we were then, Jay, we were just labeling parts of this.
Sure.
Yeah.
Yeah.
So we left at this outer area. Can you remember what that is then, Jay?
Is it the cell wall?
Not quite. So this here is the cell membrane.
Oh, membrane. Yeah. Yeah.
Okay. And
Uh-huh.
the key thing to remember is that with animal cells, they don't have any cell wall.
Yeah. Okay. Okay.
Okay, that's a key part to remember. All right. Now we're going to look at some of these other parts. What could this be here?
Is that the cytoplasm?
Not quite. I know I haven't drawn it in the best way, but this here I was trying to represent the mitochondria.
Okay. Okay.
Now, singular is mitochondrion
Yeah.
with an O-N, but plural, if there's two of them, so I can do this,
Yeah.
and I can write it as a plural, mitochondria. Okay?
Yeah.
What's this then, Jay?
So, oh, is that the actual cytoplasm?
That is indeed, well done.
Yeah.
What are these little dotted guys I put here? What are these?
They're the ribosome.
Correct. Absolutely spot on. And if the examiner wanted to be really nasty, they might put that there as well.
Is that the nucleus wall or?
Yeah, nearly there. Good application of your sense there. It's the
Yeah.
nuclear membrane.
Oh, okay.
All right. I don't think they'll penalize you heavily if you said nuclear wall,
Yeah.
but it's just a membrane. Right, good. So let's take it away and see what each of these parts do. Nucleus, what does it do?
The nucleus is in. Is it where the reproduction takes place and the?
Nearly there. It contains genetic material.
Oh yeah, contains the DNA. Yeah.
Contains DNA. Correct. Something else as well that it does. By the way, DNA. So there's chromosomes. I want you to just be aware of that word. Okay.
Mm-hmm.
Okay. Anything else? The DNA is in the chromosomes. Yes. Anything else?
I can't remember what else is it.
It controls the cell's activity. It's like the brain.
Yeah.
Activities. So if the cell needs to make any proteins,
yeah,
okay, because there's a lot of stuff in here that requires proteins to be made. DNA itself is protein, okay?
Mm-hmm.
The mitochondria, the ribosomes, they're all going to be made from proteins.
Yeah.
Cell membrane, all are going to contain proteins.
Mm-hmm.
It is the nucleus that carries the code for generating each of these different types of proteins. So the protein that will be required to make mitochondria will be different to the one that's required to make ribosomes. In other words, you can think about it from a computer programming point of view and think about it to say each of these things require different programs or software to be made. And that software is contained in the nucleus. Okay? Mm-hmm. So it contains the templates which are used to make proteins that the cell needs to function.
Mm-hmm.
Okay? Mitochondria. Is
that where aerobic respiration takes place?
Absolutely correct. where aerobic respiration takes place. Now, what about if I said where breathing takes place? Would I still get a mark?
No,
Why?
because the cell doesn't actually breathe. It's more the energy, where the energy is stored for things to happen, like reactions to take place, everything.
Ah, so you've hit something, a very important nail on the head there. Aerobic respiration
Yeah.
is a chemical reaction.
Oh, okay. Yeah.
Breathing is not a chemical reaction. It's a
Yeah.
physical action of taking air in and exhaling. So it's the
Yeah.
action of inhaling and exhaling, which
Yeah.
involves mostly gases.
Yeah.
Okay? So when aerobic respiration takes place, can you remember the type of reaction we have? Can you remember the reactants and the products?
I'm not sure, no.
Not to worry. So in this case, what we have, Jay, we have glucose.
Oh, and oxygen,
Good
maybe
man.
carbon dioxide plus water.
Good stuff. And then we get carbon dioxide plus
Plus
water.
It's just
What
the
else
other
is?
way around for the photosynthesis one.
Yay! Well done. CO2 plus water plus what else?
CO2 plus water, is it? I'm not sure, I thought it was just those two.
No. There's
Ah,
the reason why photosynthesis, respiration is taking place is not even to produce these. These just happen to be produced in the process.
okay, okay.
The main reason why respiration or aerobic respiration is taking place is to release something, to produce something. Not carbon dioxide and water. Those are just byproducts. Those are
Yeah,
not the main product. That's why you have to exhale
yeah.
the carbon dioxide, because your body didn't even want that in the first place.
Mhm.
What is it actually trying to produce when you respire?
Is it just energy?
Absolutely spot on. Top marks. Energy.
Yeah.
This is the main product you want.
Yeah.
Okay? The rest are byproducts which are not useful to the body per se, so we exhale them. This we have to inhale. This we have to ingest via digestion. Okay?
Yeah.
So where aerobic respiration takes place. Cytoplasm. Tell me about that now, Jay.
Is that like jelly-like stuff where the reactions take place?
Good man. So it's the liquid gel
Yeah.
where now these are called organelles.
Okay.
Okay, so they're supported in there. Well, the organelles are supported, but also, you rightly saw, most chemical reactions take place in here.
Yeah.
So where most chemical reactions take place.
Yeah.
Good stuff. Ribosomes.
Is that where proteins are made?
Yeah, and we can throw in a little bit of a fancy word and say where protein synthesis.
Oh, okay.
Protein synthesis takes place. Sounds a bit like photosynthesis, but
Yeah.
synthesis just means making and protein.
Yeah.
Okay, so protein synthesis, takes place. Good. Cell membrane.
Is the cell membrane, does it hold the cell together, and it's also is it like gases diffuse in and out?
Yeah,
It's
very
very
good.
thin.
So we say that this cell membrane, we actually use a word. We say it is partially permeable.
Yeah, membrane.
Yeah, it's a partially permeable. Sometimes you hear the word semi-permeable.
Yeah.
Okay, so controls what goes in and out of the cell.
Yeah.
So let's think about stuff that goes in. What goes into the cell? Give examples.
Glucose and oxygen.
Well done, good. These guys are gonna have to go in
Yeah.
so that that can carry out. Well done. Fantastic. What goes out?
The CO2 and the H2O, but the energy stays.
Absolutely, spot on. These guys go out. Now, can you think of anything else that may have to go in? Imagine you're given a 6 mark or 4 marks, and you have to give some points and some bullet points. Can you think of anything that goes in to the cell?
Not really, no.
Yeah, not really, isn't it? It's a bit of a tricky one, that one. Now, to make proteins,
yeah,
you can't make proteins just by having glucose.
Yeah, okay.
That's the reason why we have to eat proteins.
Yeah.
Yeah, because glucose alone can't sustain it. But it's something else that we're trying to get as well. And one of those things that we need that goes into the cell are mineral ions.
Okay.
Now, these go inside the cell because they are needed for the purpose of trying to create proteins. So what generally happens here, the nucleus, Jay, just to give you the full picture of what goes on, produces a template. Say the nucleus gets a message, realizes we need a certain type of protein to be produced. Now, if you remember learning about enzymes,
yeah.
So there are enzymes in the cell, and enzymes are proteins.
Yeah.
So the nucleus will say, right, I've got to go into the folder, into the library, if you like, and look up the program that produces that enzyme.
Okay. Yeah.
It then takes that program out, and then you've got this little carrier that goes through the nuclear membrane. Okay? They're called mRNA or messenger RNA. Don't worry about that now. You don't need to worry too much. But they get that template and run with it to the ribosome. The ribosome is now more like the photocopying machine.
Yeah.
Okay? Or you can think about it as a 3D printer.
Yeah. Okay.
That program is put into the ribosome, and the ribosome then produces whatever it is that is in short supply. Even if it's an enzyme, whatever it is, those will then be produced and released into the cytoplasm.
Okay. Yeah.
Okay? So that's the link there. The only other thing I just want to make you know as well is that water can move in and out of a cell. So it just doesn't go out. I know we put it as being out, but that is one thing that can go bidirectional.
Yeah.
Now, all the reactions that take place in a cell can only take place in the presence of water.
Okay.
But if there's too much water in the cell, for example, then the cell has got the ability to regulate itself and allow some of the water to leave.
Yeah. Okay.
So far, so good.
Yeah.
Excellent, good stuff. Is there anything I can add on that? Okay, so just remember that there's obviously no cell wall, all right? And these are the main features. Now, normally with a light microscope, we can see that. This you can see, and this membrane you can see. These guys here are subcellular. It becomes very hard to see. At that point, you now start needing something with a huge magnification.
Okay.
All right, great stuff. So I can take this off now, can I?
Yeah.
Good stuff. We're now going to look at plant cells. So here we go. Now we're not going to label everything on the plant cell because there's some parts that are common, so we already have done the labeling, so we're not going to worry too much about that. So there we go. My big blob there. Tell you what, I'm gonna have to put things in slightly different colors here because everything will end up looking slightly the same.
Yeah, okay.
So I'll try and add a little bit of a color scheme here to bring it to life. So here we go, Jay. We've got a plant cell now.
Yeah.
I forgot labeling this guy. So
Okay.
what's this up the outside?
The outside, is that the cell?
Is that the cell wall?
Yeah, cell wall, yeah.
Yeah, correct. Now, some students confuse the cell wall with this inside there.
Yeah.
Whenever you
Yeah.
confuse, just think about where does your wall, where do you have your wall? You have it outside your house,
Outside, yeah.
not
Yeah.
inside the house. Yeah? Okay, well, your fence is. Good. What's this inside part?
Cell membrane.
Correct. And that guy there?
The nucleus.
Good, no problemos. That thing in there?
The which thing, sorry?
Okay,
The vacuole.
absolutely. Now, normally we call this a permanent, because in plants we have a permanent vacuole. We sometimes have a vacuole in animal cells, but it is non-permanent. It may be there today. Tomorrow, if you go look at that cell, it's not there. But in plants, permanent. Okay?
Yeah.
Next.
Is that a ribosome? No. No.
I probably haven't labeled it. I'm not touching any parts here. I'm just inside this part.
Is it? Oh, you're not touching it as cytoplasm.
Well done.
Okay, thanks.
Good. Now, what will these guys be?
Is that mitochondria?
Spot on. And now we are going to have these little blue dotties. What are those?
Is that ribosome?
Correct. Absolutely correct. Spot on. And what about these little black blobs?
Would they be chloroplasts?
Absolutely. Well done. Good. Well done, Jay. Remember a lot of the stuff, actually, which is very good. Okay, so we already looked at what this guy does. We're not going to worry. Yeah, same thing as in the other instance. Oh, this here, my cell membrane. Again, I'm just going to tick off a few things here. Oops. Oh, strange. We've already looked at the cytoplasm. Anything else? We've looked at the ribosomes, haven't we?
Yeah, we looked at the ribosomes.
Yeah. And the nucleus as well.
Oh, yeah, nucleus.
Yeah, good. So all we've got to do now is talk a little bit about the 1, 2, 3 parts.
Yeah.
Okay, so what would you say about those 3 parts? What are the functions?
Is the permanent vacuole like an air space? There's air in between it? Is it to hold the structure?
Nearly there, actually, Jay. It looks like there's air, isn't it? It does look like there's air, but it's actually filled with sap.
Oh, yeah, cell sap. Yeah, I remember.
Yeah. And this provides an osmotic potential.
Oh, is that osmosis?
Keep the cell rigid.
Okay.
Or to support the cell, shall I say.
Okay.
So if the cell sap left, this will shrink and plasmolyze, okay? So this will have a certain amount of water of a certain concentration, which allows this to push out and keep the cell firm. Okay?
Yeah.
So cell wall, what does that do?
Does that hold the structure?
Yeah, or provide support.
Yeah, provide support. Good.
Holds it, you're right. Good. What's missing? Chloroplast.
Is the chloroplast that will do where photosynthesis takes place?
Yeah, so this contains chlorophyll.
Yeah.
Good.
Which makes the plant green.
Absolutely contains chlorophyll, which absorbs sunlight.
Oh, yeah, yeah. Because sunlight is,
Photosynthesis.
yeah,
Well done.
is needed for photosynthesis.
Absolutely. The question is, do all plant cells have chloroplasts?
Yeah, because,
Okay. Let's think about, oh, now it's saying my internet connection is unstable. Oops, I hope it doesn't play up too badly. Let's think about root cells, root hair cells.
root cells, okay, yeah.
Would you expect them to have chloroplasts?
Oh, root cells wouldn't need it because they're in the ground. They just have a high surface area and they're absorbing water.
Very good, and very good for remembering that concept of surface area. It's very big in biology. Yeah. Good stuff. So not all cells will have chloroplasts in them.
Yeah. Okay.
All right. That's a key thing to remember. So one thing the examiners like asking is the differences between animal and plant cells. They love that. The differences between animal and plant cells. So the key thing is, these are the differences.
The cell wall would be one difference.
Yes.
Yeah.
2.
And the vacuole.
Permanent vacuole. Good.
Yeah.
And another thing I want to just make a note while we gradually come to a close of our session is the general terminology that we use to call animal and plant cells. We use one word which describes or to talk about both animal and plant cells. Do you know what that word is?
Would it be specialized cells? Or?
Ah, okay. You're kind of thinking there in terms of a next layer that we will be going to. So you remember a lot, actually. But this term is the term eukaryotic.
Okay. I think I've heard that.
Yeah, I think many moons ago.
Yeah, I remember some about it. Yeah.
So animal and plant cells are described, both of them, as being eukaryotic.
Oh, yeah, I remember that.
So anytime you hear in your biology paper, draw the structure of a eukaryotic cell, you can draw either an animal or a plant. Okay? Yeah. Plant or animal cell. Now, eukaryotic, sometimes you hear the word eukaryotes. Eukaryotes is simply describing a species that has eukaryotic cells.
Yeah.
And we say that eukaryotic are multicellular species. Meaning they've got lots and lots and lots and lots of cells that are all specialized to do different functions.
Yeah.
So eukaryotes are not found as a single cell. They're multicellular.
Yeah.
Okay? So that is a feature of eukaryotes. They are multicellular species.
Yeah.
And the cells in them are specialized and differentiated, if you want. You can put that.
Yeah.
So far, so good.
Yeah.
All right, good stuff. So I think at that juncture, I won't go so much into specialization and differentiation, but we might touch on that a little bit, and just to build you up and make sure all the basics are right, and then we'll have a look at trying to look at some of the other stuff as well that you may be doing. I think you did mention diseases as well, transmitters of diseases, one of the things you wanted to cover.
Yeah.
Cool. We can keep that on the cards.
Oh, yeah, diseases, yeah.
Any questions for me, Jay?
No, I think it's all good, yeah.
Good stuff. So I'm going to send you a little document that you can use just to brush up on anything. You don't have to do the whole lot, but especially if there's anything you think, oh, I haven't done that in a long time, let me brush up on that, then do attempt it by all means, okay?
Yeah, okay.
Good stuff. Fantastic. Yeah. Well, Jay, the session definitely has come to an end now. So what I'll do is we'll meet up again in the week. Just trying to think. When last did we meet, Joe? Was it Sunday?
Sunday.
Was it Sunday last week, yeah?
Yeah, so we're only missing one session.
We're missing one session, yeah. So we
Yeah.
can have that session on Wednesday, perhaps.
I have football training on Wednesday.
Okay, Tuesday.
So Tuesday, yeah, I think it should be all right. But could you do Thursday?
Thursday. I could do Thursday. Thursday, even though it's going to have to be a late start from about sort of 5:30.
Yeah, that's good. That's all right. Yeah, because that'd be
Cool.
probably better for me because I get back at like 5 from school. So
Oh, okay, so you got
that'd be
half
all
an
right.
hour to catch up. Yeah, all
Yeah,
right, that's fine.
exactly.
Get a little bit of a break
Yeah,
before we start. Okay, no problem, Jay.
yeah,
So I'll pencil you in for Thursday, 5:30.
yeah.
Good
That's
stuff.
good. Yeah.
Nice one, Jay. Have a good evening, yeah?
Yeah,
And I'll
and
see you
you.
Thursday.
Thanks for the session. Cheers.
Welcome, Jay. Anytime. Take it
See ya.
easy. All right.
Cheers.
Good, right.